All right.
Good morning, good afternoon, dear friends, colleagues, and viewers. So it's my pleasure today to welcome Dr. Sherif Abouelhadid at the channel, Innovative Sustainability channel. So before he will begin his presentation on the vaccine, his studies on vaccine, conjugated vaccines, I'd like to have a brief introduction. So Dr. Sharif has started his education in Egypt at the Misr University of Science and Technology, specializing in pharmaceutical sciences and drug industry. He followed into Alexandria University, where he received a diploma in biotechnology, and then he received a PhD degree at London School of Hygiene and Tropical Medicine, University of London, specializing in pathogen molecular biology. So it's my pleasure to welcome you today. Dr. Sharif, welcome, and the floor is yours. Please start.
Yeah, thank you very much, and thank you very much for your invitation. When I first came across your channel, I was so excited because it was about right time that actually someone actually start a channel discussing discussing this very important topic. And thank you so much for your invitation. So I'm going to talk briefly about conjugate vaccines and about why do we need conjugate vaccines and a little bit about. So I'm not going to talk about actually my research. I'm just going to talk about what's going on because I don't want everything to be around me. Just basically, we are actually facing a bigger problem, which is antimicrobial resistance. Um, so in brief, actually, so I'm just going to give a little hint of why or where actually my interests in conjugate vaccines were developed. So after my graduation from School of Pharmacy, usually actually after I graduate from School of Pharmacy, especially in the Middle East, North Africa, is you have two ways, either to work as a medical rep or basically you work in a community pharmacy. And I wasn't really interested in both. I was really interested in science because in my fourth year, when we started actually learning about molecular biology and DNA technology, I was really hooked, as you can say. So I really wanted to pursue it. So basically, I started working first in a lab, in a virology lab, and then I moved to a big pharmaceutical company in Egypt. This is where I was introduced to conjugate vaccines. And little did I know that actually that antimicrobial resistance is causing a big problem around that time. That was around 2007. So the talk
Yes.
about antimicrobial resistance wasn't actually that big, but the data was there. That's the funny thing about it. And then I started working on conjugate vaccines, and then I was working developing vaccine against Salmonella typhi. Of course, because young me didn't know much about actually that Salmonella typhi is still causing problems because
It
when
does.
you
It
talk to
does.
actually to many scientists and you tell them, for example, oh yeah, well, typhoid fever, they go like, is it still causing disease? Some of them actually ask this question, non-microbiologists at least, and non-epidemiologists, of course, and non-infectious
Mhm,
disease.
mhm.
So I started working on it, and then I was working on chemical conjugation, which is very very elegant platform. However, it's quite complicated, and I will talk actually why actually this is basically I find it quite complicated and complex. It works. We know it works. There are vaccines actually in
Yes.
the market that
Yes. It's
are
hard.
developed by chemical conjugation, so we know that it works and it saves lives. Okay, so that's not to basically say anything about that. So anyway, so around this time, I was studying actually my diploma in Alexandria University, and because actually when you in your master's degree or maybe on your postgraduate degrees, everyone will start talking about, oh well, we need actually like more enzymes to simplify
Mhm.
the chemical processes because actually chemical process actually takes time. You have products that you need to purify, and that adds to your basically your expenses on the production. And this is basically when I first actually starting looking at an enzyme that can basically simplify the chemical process in order to develop vaccines. And this is when I came across Brendan Zren's group at London School of Hygiene and Tropical Medicine. I wrote to Brendan, who was generous enough to basically to offered me to come and visit for 1 year. I went there for 1 year. During this time, a revolution happened in Egypt. So basically, I never thought about
Yes.
that. And then one thing led to another, and I stayed in the lab. I did my PhD, and then I returned back, almost returned back to Egypt. So it's basically 80% of my time now is in Egypt, and 20% of my time is in Europe and UK. So I will share some slides. So basically, to to actually to give you an idea why this is, why do I think actually conjugate vaccines are important? Why do I think that actually that the technology that I was part of developing at some point in my life is also, among many others, of course, I didn't develop it myself.
Yeah, while you're uploading, and just
Um,
for the viewers, I mean, those who are savvy, they understand, but those who are new to conjugate is basically the efficacy of the, how to say, success of the chemical conjugation. You always get a very low, like it's about 5 to 7%, and it's a lot of losses. So, yes, you have a reaction, you have ingredients, you have protein, and for example, polysaccharide, but the actual product is really low yield. So that's why not efficacy is wrong word. It's a yield. The yield is low. So of course, the reason you're moving to enzymes, it's very,
mhm.
it's understandable to look for better solution for less losses during the production.
Yeah, thank you very much for pointing that out. That was that's very important. So actually, so the thing is actually, so I moved from the UK, and actually I took a bit of a step outside academia, uh, or actually to put a foot outside academia and another foot inside academia. So I'm still partially doing some academic research as well. But the idea why I took this, quite what I see actually as a quite courageous step or daunting step, is for a very simple reason. Is because when you work a lot in vaccine development and you work in academic setting, you need actually to understand how the market works. And that's actually, that side is quite missing sometimes in academia, because we usually talk to ourselves. In our conferences, we talk to ourselves. We don't talk actually many to pharma companies, and actually, and for some reason, it's really sad that actually some actually see pharma companies not in a good light. So what I'm doing now is basically I'm managing director in a biotech consultancy company. It's called Biodar. It's in the UK. And I'm also director at Perant Foundation for Science and Philosophy, is a non-profit organization that aims at expanding the landscape of biological sciences in the Middle East, North Africa region. So let me jump straight. Why now vaccine development is really important, and actually because it was actually important from the start, to actually from the beginning of the first man or first human being who basically contracted the disease. But after COVID, actually, I think most of us kind of faced a harsh reality. And this harsh reality is there is vaccine inequity and there is vaccine inequality, and it's real. And it's devastating, and many lost, actually many deaths caused by something like this, because vaccines were simply unavailable or inaccessible.
Yes, correct.
So, for example, this paper here was published in Nature Communications 2023, and figure A here shows that actually that the countries modeled according to the WHO region and the percentage of fully vaccinated individuals as of October 2021. And you can see, for example, Egypt has around 6% of population actually got vaccinated. However, actually, for example, other countries got even more. So what did this tell us? That basically that yes, we are facing a big of a trouble that actually, and we've seen this as well, because when you have countries that are unvaccinated during pandemic, actually they could be even like a hotspot for the next variant that's going to come, right?
Yes, it stays long,
So
so it can
that
mutate. Yeah, absolutely. Yeah.
100%. So that means that actually that this is basically a global imperative for vaccines to be available and accessible. And this here as well, it shows you that actually, for example, you can see even in 2021 and 2024 and April 2021, you can see that basically that the fully vaccinated per 100 population on the y-axis in high income countries in yellow is quite increasing. However, actually, for example, you can see even around a couple of years later, low income countries didn't even match up. Low to middle income country in blue just basically reach the same, for example, percentage that the high income countries reached in 3 months. They reached it in around, I would say, almost 9 months.
Mm.
Okay. So that tells you that actually the low to middle income countries in blue, the high income countries in yellow. So that tells you
Mhm.
that actually that this is serious, right? And we saw that actually during the COVID pandemic, countries actually basically, some countries kind of like shut down on their own stock of vaccines. So they didn't share it. And I'm sure that actually that there were funny other political games played. And you can, if anyone interested, they can read The Guardian and they will find plenty of them. All right. Figure B here shows that actually that, for example, uh, if the population in low to middle income countries and low income countries were vaccinated the same as the United States, how many deaths could have been averted? And for example, in Egypt, 16,000 deaths could have been averted if they were actually vaccinated the same as America. Some are quite high as well. For example, such as Bangladesh, it's almost 93,000. Okay, Indonesia around 150,000 as well. So that's these are numbers, and the way that I see it is basically this is not only numbers and human lives. It's basically that's something related to economy, families that they lose their loved ones, and this is all I would say could have been averted. And you know what they say basically is one death is a tragedy, thousand deaths is basically is statistics, and that actually has to
Yes,
be changed.
yes, yes, you're right.
All
And
right. So basically,
every death is basically at least three people, four people behind the person who actually suffering. So it multiplies.
yeah,
It's
yeah,
at
hundred
least,
percent.
yeah,
At least, yeah.
at
So the
least,
shocking
not.
numbers, yeah, yeah. So the shocking numbers that we're facing, it's just basically everyone's talking about AMR. The paper published in Lancet that basically AMR caused death around 1.2 million, caused a direct death of 1.2 million deaths globally in 2019, and basically it kills more than HIV, breast cancer, malaria. And if this going on actually at the same rate, same pace, this could lead to between 10 million deaths and in a more pessimistic
Mm,
studies around 39 million deaths in 2050, and that might be causing around 66 trillion pounds of loss in the global economy. So this is real, and this is basically a big problem that we're facing. And this has to be communicated also very well because actually some policymakers still didn't actually see how devastating the situation is. So is it new? Well, the bacteria becoming resistant to antibiotics? I don't think it's new at all. I think it's just I always think that actually, and this is something that I teach to my students in class, the bacteria will always resist antibiotics. It's just basically we try to prolong the time where actually the bacteria is going to resist the antibiotics, so basically can develop new things and other things. And one of the first actually scientists who ever discussed this was Alexander Fleming himself. He mentioned that actually, like maybe a thoughtless person not taking the antibiotics in the right way will actually lead the resistance. And you can see here, for example, this figure shows the timeline of developing or actually or discovery of antibiotics and their resistance emerged. And that's actually, for example, penicillin. It was around 1928, and then the resistance appeared in 1940. And that was actually quite new. And then you can see this a lot happening a lot in different antibiotics.
Yes,
Okay,
yes.
so can we develop more antibiotics? The problem is no, because actually, or we hope, but actually, but the problem is there is a discovery void that's quite clear that actually that very few antibiotics or almost none, or basically handful that we can count on one hand that's been actually on the market. The reason why different things, there is no financial incentives. Antibiotics also basically are given for specific course of time, unlike chronic disease. So, for example, if I'm a pharmaceutical company, I want my drug to be used over and over again, other than just
Yes.
basically 1 week. And there are no easy targets. And that leads that actually antibacterials basically capture around 5% of the total venture capital of pharmaceutical research. So the gap between development and sales is enormous. And that's something that's a reality that that we hit hard. So let's talk about what are the alternatives. Of course, we can use bacteriophages and something that actually that I'm interested in, but then I will talk about vaccines because vaccines could help and protect many people. So carbohydrate vaccines, in theory, just think about it. Actually, all these yummy donuts, and it's about lunchtime actually in Egypt, so basically I'm quite hungry. But actually, all these yummy donuts are basically you cannot see the dough of the donuts themselves. The reason is why? Because actually they're covered with sugar, they're covered with icings. Same as bacteria. So
Mhm.
basically, you cannot see the surface antigen of the bacteria because actually they are covered with icings, and this icing is made of carbohydrates. Many years ago, a brilliant scientist, his name was Aldous Avery, he isolated this sugar coat and basically injected in lab animals, and he found that it elicits immune response, but it elicits um, what we know later is T cell independent immune response, which is basically
Mhm.
short term memory. And then he, in 2000, I mean 1931, he basically conjugated this carbohydrate with a protein, and he found that actually this elicits long-lasting immune response. In my in my humble opinion, this is basically a groundbreaking discovery. Um, and actually, uh, and it's funny enough because actually the same concept stayed like until nowadays. So this is a timeless discovery. So later we knew that actually polysaccharide themselves or the sugar coat itself, it basically elicits T cell independent immune response, which means that you will have short lived antibody production because there is no memory cells, and then you need this interaction between B cells and T cells when you conjugate this sugar coat to a protein that will elicit T cell dependent immune response. And that's basically the T cell and B cell interaction is very important for developing a long-lived antibody production and memory cells. So that's why we do
Mhm.
conjugation. Okay. So the way that we used to do conjugation is through chemistry. It's elegant, but it's believe me, it takes long time, and it's laborious and it's expensive and need a lot of purifications, need lots of no chemistry knowledge as well. And some of them are random conjugations, so you don't know actually where basically the glycan is attached. Basically, it's because you activate amino acids on the protein, and basically you conjugate you add the
You're right. It's a good
polysaccharide.
point. It's not precise. It's not precise,
Yeah,
and
yeah.
every time you have to basically confirm that conjugation did take place, and this O-link or whatever you use is there. So, like by mass spec or any other,
Yeah,
it takes
exactly.
time.
Yeah, yeah. Some methods that
You
were
have to
basically
prove.
were trying to be more specific, but it's just basically,
Oh, true.
well, you know how chemistry works. It's just,
It's
it,
length. Also, length is different, so it's
yeah,
not the same.
yeah,
Not uniform, I
100%.
should say. Yeah.
Yeah, exactly. So basically, in 2000, in year 2000 and in 1999, basically, Brendan Ren's group in 2000 actually they were doing the sequencing, or they published the sequence of Campylobacter jejuni, and they found that actually that this bacterium, for some reason, they don't have all this type 3 secretion system, all the sort. But actually, what they found this very interesting locus, which is basically has this particular enzyme, it's called PIGL-B, which looks similar to the STT3 in human. So basically, what they found is basically a very interesting enzyme in a polysaccharide or a glycan biosynthesis region, and that there is central enzyme that actually that later, after many biochemical assays and genetic knockouts, found that actually that this enzyme can catalyze the transfer of glycans to proteins. And it does this in Campylobacter jejuni. So of course, knowing that actually that any scientist will do, anyone basically interested in translation science will express this enzyme in an E. coli with a polysaccharide and a carrier protein, and then look where actually basically whether this enzyme can catalyze the transfer of this sugar that they want, the desired sugar, the antigenic sugar, to an immunogenic protein. And that's basically what the technology is all about. Okay, so this is basically the difference between both. This is basically on the left is chemical conjugation, on the right is bioconjugation. And why this is important? Because we were just discussing. Basically, I remember it was couldn't say it, but basically it was actually something big that basically you grow pathogenic bacterium in fermenters and you isolate the sugar coat, right?
Oh,
And
true,
then you
yes,
basically
of course. Yes.
and that was scary in itself. Just imagine that you're passing by a fermenter, someone told you there's salmonella typhi in there. That's a nightmare for a microbiologist.
Worse than that, could be diphtheria, but it's not the topic here, but nevertheless, there are still vaccines that you grow from the pathogen, and they purify from pathogen. So it is, and from the biohazard standpoint, how you utilize and treat that, it's a lot of work on the actual waste management after. So you're right. If you can avoid it,
Yeah.
that's already sustainability.
Exactly, yeah. So basically, you grow the bacterium in fermenters, and you grow other bacterium because it's a carrier protein in the fermenter. And you basically do lots of purification, you do lots of chromatography, which basically adds more money, which basically that's why it's have the dollar sign here. And you couple them together, you activate them, you couple them together. Once again, you have also lots of byproducts that you don't want. So basically, you keep doing um, a purification, and then you do later characterization of the final product. And then when you reach the final product, you spend a lot of money, and many scientists who've been working on it. So basically, and it's quite lengthy in time. So, and expensive, it's costly. So basically, that's why you end up by a product that's quite expensive in a way. Sometimes it's unaffordable for low to middle income countries to buy it. So the alternative is basically to try to express everything in E. coli and then let the E. coli to work as a microfactory for your vaccine. So basically,
Mm.
the E. coli has the polysaccharide suppressing plasmid, the protein that is going to the immunogenic protein, and then you have the enzyme that basically couple the transfer of the polysaccharide or the sugar to the protein, which will basically lead to eliciting long-lasting immune response and memory cells. So this basically is so the chemical conjugation is basically simplified more in biological conjugation, and then you do some purifications. Yes, you will have to do some purifications, of course, but they will be far less. And you know actually that basically that the glycan is attached to a certain specific amino acid after doing mass spec, but basically usually is because actually where it binds, where the enzyme actually transfers the glycan to a specific amino acid. So so the product is easier to be characterized, if you can say,
You have
right?
more targeted approach where to look. Yeah, you're
Yeah,
right. More
exactly.
targeted.
More targeted approach rather than random approach. And I know that this figure is just basically this slide is a lot of things, but this is to show you that actually that this technology helped in developing candidate proteins again, conjugate vaccines against different bacteria, Streptococcus pneumoniae, aureus, Shigella flexneri, Shigella sonnei, Salmonella enterica paratyphi A, Francisella tulurensis, Klebsiella pneumoniae, and so on. And many other actually enzymes been discovered now. Not only the one that was found in Piggle B, other one was discovered in Acinetobacter baumannii, and another one was discovered in Neisseria meningitidis. and another one Neisseria gonorrhea. So it's basically
Mhm.
the idea of actually basically hunting for this magical enzyme that can catalyze the transfer of a glycan to a protein to basically simplify and reduce the cost and the time as well that's used to be taken by the chemical conjugation is I find it fascinating. So now actually there are some answer questions to be answered, and they are more technical questions. So is it feasible, for example, are pharmaceutical companies excited about actually about this technology as much as academics are excited about it? That's actually and there's been always a discussion. You find actually you go to a conference in academia, everyone is excited about this technology, and when you talk to industry, they are least excited about it.
Mhm.
And the other question, another technical question, what's the best model to design a bioconjugate? For example, either to basically to add it as a single in the tail of the protein, for example. So basically you have a linear polypeptide and then you have the glycan attached to it, or basically you attach it in on the protein itself. And that attaching it on the protein itself, which is on the left, just basically it requires more of engineering because you know the protein structure and you don't want to basically disturb the protein or any
Okay.
epitope. So that's actually something technical as well. Other technicalities are basically the enzyme specificity, this magical enzyme specificity. You need to improve it, you need to increase it, you need to alter it, and so on. And can this enzyme be simplified? The reason for this is because actually this enzyme, just we take it from Campylobacter and it has, let's say, different domains that you might not need. Can we simplify to make it even better enzyme? Because when you express it in E. coli, it's quite toxic. So basically, can we use a better enzyme and simplify it? Could AI actually help in advancing this biotechnology? Everyone's talking about AI usage now. So can it help in biotechnology? These are some technical questions. But the bigger questions that actually that also that we need to discuss actually, can this biotech actually decentralize vaccine manufacturing and reduce vaccine inequity? Can it basically solve this problem providing accessible vaccines and affordable vaccines? What are the infrastructure required to adopt this technology in LMX, knowing that actually that this is not basically you grow cells in fermenters and so on. You need some other sophisticated actually methods in order to basically do the genetic engineering of the bacterium, which is going to be your microfactory. What are the frameworks for intellectual property and knowledge transfer essential to foster an equitable R&D ecosystem in LMX? And this is not related to this biotech in general. It's just basically related to vaccine manufacturing in general. What are the intellectual property and knowledge transfer essential to basically to have I wouldn't say equal, but like half matching at least
Mhm.
R&D research in LMX. And how can Africa leverage regional hubs to transition from fill and finish to end-to-end vaccine R&D? And these are the questions that basically that we ask a lot, as you know. And by this, I finish my presentation. I'm happy to.
Thank you very much. I think, yes, I also looking, we're kind of, close to the running out of time, but I think we still have a minute or two. I
Mhm,
would say that questions are critical, and from what I heard from you, and, I think also where this more miniaturization of production going, make it more personalized or make it, achievable for many countries to
mhm,
adopt is basically the model that you're suggesting with E. coli and having both produce the protein that the carrier and also the
mhm,
polysaccharides and have conjugation. Presumably, if you have this pilot study, and I imagine that's a goal for the
mhm.
company for Biodar, is to may scale up, scale up
Yeah.
to not so much maybe volume wise, although that also could be one of
Mhm,
the on your agenda,
mhm,
but basically propagate as these small plants in other
mhm,
countries. And maybe it's government to government initiative, or maybe some
mhm,
through the the organization that you're working with. a philanthropical path, but still
Yeah.
government people need to be
Hmm.
involved because this is a lot of
100%.
regulations always, right?
Hmm.
But that could be the path to bring people on board. And I know African countries now have their own regulation
Hmm.
for drugs, so that could help to channel this initiative. But that definitely initiative is worth pursuing. And when you said that pharmaceutical companies kind of like looking puzzled that you or not what, but they are usually need to see the production happening before they
100%.
like seeing like benefits or advantages,
Yeah.
economical advantage, and so forth. So it's always hard to bridge that link. But I think if
Hmm.
you have alternative path pushing forward, that can help
Hmm.
a great deal. And definitely this presentation is kind of fresh air, new idea, but new idea
Hmm.
that you already have as the
Yeah.
pilot working. And
Hmm.
yeah, definitely it's a call for action for viewers who those interested to contact Dr. Sherif and see where it can be implemented. But not just also
Hmm.
African countries, I would say many other countries who would be interested in the technology to
Yeah.
pick it up
Hmm.
and see what's
Yeah.
possible.
Mhm. Yeah, I think you're right. And the beauty of this actually technology is basically it's more like a plug and play. You can change actually, you can change the protein that you want. You basically can add another protein, just basically add the sequence or the tag where the guy can be attached, and you can have another uh, candidate vaccine.
Sakurai to another
Uh,
candidate, yeah.
yeah, and the point that actually that I'm sure that you know, which basically is because for example, Streptococcus pneumoniae vaccine has been developed actually many serotypes or some serotypes in the vaccine are not present, for example, or in Africa, and the ones that are causing some disease in Africa are not, for example, in the vaccine. And this is actually that can cause a bit of a problem, right? So this can allow um, countries to develop their own vaccines, hopefully. And I think actually, I think in the discussion as well, that actually that I see it a lot between industry and academia in governments as well. That basically that after COVID, many countries actually kind of like it was sobering to see that no one can save you
Yes,
unless
yes,
you're going to
yes.
save yourself.
It's a sobering thought.
Um, it was it,
Self-sufficiency.
yeah. Yeah, because when some countries actually having their own problems, they don't have the time to save you. And low
Yes.
middle income countries actually started to develop their own technologies. But I think not only this technology, not only the bioconjugation technology, but I think actually many other technologies, vaccines, can play a role in at least or sow the seed that basically that you have a more bigger vaccine or active vaccine manufacturing and research as well. So yes, BioDarwin is basically working on consultancy, but if anyone is interested, we're happy to basically to contact
Oh,
and work with
definitely.
them.
That's
Yeah,
very interesting, and even for this area, for this region, definitely there is at least an opportunity to discuss, and
100%.
I think what I can do from my side, I definitely will. But I think it's cool also for viewers across the globe because people who are watching this channel, they're in Canada and US as well, and some in
Mhm.
France, and also a few started to join here from this region. So
Mhm.
before we run out of time, I would like to thank again Dr. Sharif. It was a pleasure to
Thank you
have
very
you
much.
on this channel. And also the topic is really thrilling, very interesting. So thank you again for joining, for finding time and for joining
Thank you
and
very much.
wishing all the best. And hopefully we can, meet again for another
Sure.
presentation. And yeah, thank you very much. Thank you.
Thank you very much. Thank you for the invitation. I will hope to see you soon. Okay, stay safe.